Hey guys, so in this lecture we're going to discuss chapter 13. We're going to discuss the ideas of sterilization and disinfectants along with the concept of antibiotic therapy. So let's go ahead and get started. So there are some basic terms that we need to understand for this lecture. There's sterilization, disinfectant, antiseptic, and sanitation. So sterilization is whenever all living spores, viruses, everything is destroyed. Disinfectants is where you remove or kill the disease-causing organisms. And then antiseptic is very similar to disinfection, but it applies to removing pathogens from a surface of a living tissue. So a different example between disinfectant and antiseptic. So disinfectant would be like you wiping down your bench top, and then antiseptic would be you wiping down your body for like a surgery or something of that nature. So disinfectants has to do with inanimate surfaces such as like toys, chairs, hairbrushes, stuff like that. And antiseptic has to do with live tissues such as like a patient. And then sanitation, this is where you are just simply reducing the microbe population to a safe level that you can interact with. It also involves primarily dealing with inanimate objects. So you don't really sanitize your body. You normally sanitize a nipple of a bottle or something of that nature. So definitely know the differences between those guys. And then with medications, there are two main classes of medications that we are going to discuss. There are cidal agents and then static agents. So cidals, if you have a medication that ends in cidal, then that means that it kills all the microbes designated for that pill. So if you were to take an antibiotic that ended in cidal, then it's a bactericidal. Whereas if you had a fungal infection, you take a fungicidal. So the idea for cidal drugs is to completely drop the number of microbes present. So let's say that you had an infection, you feel sick, then all of a sudden you go to the doctor, they prescribe you a medicine that is designed to kill all microbes, then you would see over time that there is just a plummet of microbe growth and it's non-existent. Whereas a static drug, what it does is that it will somehow inhibit or control the growth of the microbe so you can actually have an immune response to it. So the idea is that you have the infection and then you're taking the medicine, and then slowly but surely you'll have stagnant growth in it. So that's the same concept of where you want to limit the growth, but instead of actually killing the microbe, it will just contain the microbe. Okay, so be able to definitely recognize between these two graphs as well as the concept between cidal and static agents. Okay, so there are several factors that will influence the speed of which there is a lethal dose of medication. So some factors are what was the initial population size? So theoretically speaking, that if you have a larger population, the longer it's going to take for it to decrease. And then also, what is the population composition? Is it spore-based or is it just cell-based? If it's spore-based, it's going to be a lot harder because spores are a lot more sturdier. As well as the concentration of the medication, the duration of which you were exposed to something, and then as well as that, where is it present? Is it present in blood? Is it present in feces? So certain things like that can definitely alter the effectiveness of a drug or damage to a host. So this is kind of just an idea of ideally of how effective do you want a drug to be. So the idea is that you want 90% of your bacterial culture to be dead at a certain time point. But honestly, on the test, guys, I'm not going to ask you the curve or anything or to calculate anything. I just want you to know the idea of it. So ideally, you want, after a certain amount of time it takes for a medication to work, you want it to ideally kill up to 90%. So in this image, it shows right here the 90% is roughly just before the 5-minute mark. So that's pretty effective. So you do ideally want a drug that is very, very quick, but you also don't want it to be too quick where it can actually harm the patient. So I just want you to know the theory behind it. Ideally, you want your medication to be able to kill 90% of the population within a good time frame. And also the idea of like this overkill, like you want to kill so much, is so it's precautionary based. It's more like preventative, so like it doesn't actually come back with the vengeance essentially. So you just want to make sure that there's minimal to ideally no presence of bacteria for your patient, so they're not getting sick later on from the same microbe, and that same microbe actually may even develop resistance. So that is partly why you always want to take all your medication, like your antibiotics, for the 10 days that it says and not stop at day 5, because it can actually create resistance. So always take your medicine, guys, and make sure that you're taking it effectively. Okay, so can a microbe be controlled without chemicals? And the answer is yes. So the idea is that you want to kill them or like sterilize them or clean your cultures or anything of that nature. There are several ways that you can do it. The first one is temperature, and then there's pressure, filtration, and eradication. So temperature is whenever you literally just cook them. You just cook them, and then with heat, a lot of times, if you heat them up, you're killing it. If you're putting them in cold, you're inhibiting it, and then you're actually preserving them. So they kind of like the ice age for them, like they kind of just go dormant. Or there's pressure. A lot of times the pressure is combined with temperature, and that's called autoclaving. So autoclaving is the idea of sterilizing your culture. So that's actually what happens to our cultures whenever we're done with them. They get sterilized. So they get put into this autoclave so they are completely dead. So there's increased pressure and increased temperature, and there's like almost no microbes that at least we work with that can withstand that, and then they die off. And then filtration, it's used because chemicals they could be temperature sensitive. So this is whenever you are just literally pouring them through a filter. So you have a culture, you filter them out either through some kind of like high grade strainer essentially, and then you just pour your cultures through, and then the idea is that that filter is catching all your bugs, and then you get a clean culture. And then there's eradication, which is where you pretty much do chemotherapy on them. Okay. So the effectiveness of a given chemical agent depends on a multitude of things. So it depends on if there's presence of organic material, what kind of microbe you have, if it's corrosive, and then the stability of your surface. So with the presence of organic material, a lot of times chemicals will bind to organic materials, so that will lower its effectiveness against microbes. So ideally you don't want to use something that's very organic based. And then also the kind of microbe. So ideally it should be effective between a broad range of pathogens, so you're not just so narrow. So you want to use something that can kill multiple species rather than just one species or just one type of microbe. So you want something that kills viruses, bacteria, and fungi, but you don't want something that only kills viruses. So you want something that's a lot more broad spectrum. And then corrosiveness, I'm not really too concerned about that, but the idea is that you don't want your cleaning agent to destroy your surface, pretty much. And then also, like, how stable is it, and then does it smell? Like, how long can it be on the shelf? So ideally, you want it to be stable and not be a gross smell like bleach. To me, bleach is absolutely disgusting, but it's really stable on a shelf. Same thing with like Lysol. Same thing. And you don't want something that has high surface tension, meaning that it won't adhere to your surface for long periods of time. So that's the idea behind disinfectants. Okay, so there is a test. I won't ask you to do this on the test, but I do want you to know the idea of it. So the phenol, I won't ask you to calculate or anything, but I want you to know the idea of the phenol coefficient test. So this involves whenever you're inoculating a certain number of bacteria into a diluted disinfectant agent. So with that, what you'll do is that you will calculate the coefficient. So the phenol coefficient is based on the highest dilution, which means that it's the lowest concentration of culture that will kill the bacteria after 10 minutes of exposure. So the higher the coefficient, the better efficiency. Okay? So such as right here. So like these guys right here in the red boxes, this is perfect. So at 5 minutes, like there's still presence, but it also has the lowest concentration because it's only 100 as compared to 200. That's pretty dilute, whereas 1 to 100 is not as dilute, so it's 1%. So you don't want so concentrate, but yet at 10 minutes you want it to be 0. So that's the idea behind. So like if on a test, if I were to ask you, these red boxes would be a way, and then I would ask you to like box which of the disinfectants is the best. But I won't ask you on the test to do that. I just want you to know the concept. So the concept is the higher coefficient of phenol, so is the better efficiency, and then also with that you want to know what of your cleaning solution has the highest solution, but that is still super effective after 10 minutes. So that's the idea. Okay, so there are a lot of disinfectants listed on here. I'm not going to read them off, but these are just some of the highlights as to like what is their mechanism of how do they actually control or eliminate microbe. So I'm just going to point out some of them because some of them we actually use in class. So the main one that we used in class is alcohol. So like we have the ethanol squirt bottles in our lab. Specifically what this does is that it will disrupt the proteins and the lipids of the bacteria, so like on the cellular membrane, so it will completely denature it, and it literally just makes them fall apart and melt. So I'm just going to highlight that one, but if you want to read through them, pause the video and go for it. Okay, so now we're going to get into some antimicrobial therapy concepts. So ideally, antibiotics, you want them to be selectively toxic, meaning that you only want them to kill or inhibit the pathogen and not hurt your patient. So there are a lot of antibiotics out there. Some of them are considered broad spectrum, and some of them are considered narrow, as well as being bactericidals and bacteriostatics. So as you see on the table below, like penicillin itself is pretty narrow because it only can kill gram-positive bacteria. Whereas tetracycline, it spans across 3 different types. So penicillin compared to tetracycline is the broad spectrum, whereas penicillin is the narrow. Okay, so that's just a concept that you want it to be selectively toxic, meaning that you want it to only hurt the pathogen and not your patient. Now the actual test in order to determine what is the minimal amount of medication that needs to be present, it's called the minimal inhibitory concentration, or MIC. So MIC is defined by using the lowest amount of drug, but where it is still effective in preventing microbial growth. So that's just the idea. I'm not going to ask you to calculate or anything. I just want you to know the concept. So the concept is MIC. So minimal inhibitory concentration. So this is where you have the minimal amount of medication that will still kill the microbe. Okay, so next. So on the test, I will ask you something about a susceptibility test. So this is where, like let's say you have a skin infection and the doctors don't know what to do to treat it. So in order to treat your wound the best they can, what they'll do is something called a disc susceptibility test. We'll actually do something like this in lab, but not with antibiotics. We'll do it with disinfectants, but the idea is still the same. So what you do with this is that you will plate a garden of the bacteria. So you'll get a culture from your patient, you will streak an entire plate, and then what you'll do is put these little discs throughout the entire plate. And then from there, after it's grown for maybe like 48 hours or so, you'll come back and you'll see plates like this where they have halos. So these halos are called zone of inhibition. So from there, what you'll determine, as you see, I'm going to talk about the right one because it's a little bit easier to determine. On the right one, you can see that there are several different halos, but they differ in size. So ideally what you'll do is that you'll measure each zone. So you'll measure the diameter across of the circle, and then you'll compare that. And then whichever one has the biggest zone of inhibition, that is the medication that you should give your patient, because that is the medication that has shown to not allow growth of that microbe. So if you were to see something like the OX one where there is no halo, you would not give your patient that drug because it shows not to be effective in inhibiting the growth or killing the microbe. So on the test, I'll definitely ask you to know the concept of it and then also be able to determine which one is the best one to give based on the zone of inhibition. Okay. So now more concepts about antimicrobial therapy. There are two different types of drugs and how they work. The first one is called synergistic. So the idea of if a drug is synergistic, that means that it needs to have another drug present in order for it to be effective. So synergistic, so if you give one drug, so if you give drug 1 by itself, it won't work. But if you give drug 1 on top of drug 2, then it works beautifully and your patient is recovering. So that's the idea of synergistic. Now we have antagonists. This is where drugs will interfere with each other. So this is where if you give drug 1 and drug 2 at the same time, they kind of cancel each other out, or one of them gets diminished. So I have an example right here of penicillin and the macrolides, but another one that is probably more common that you know of is birth control and antibiotics. So if you are on birth control and then you take an antibiotic pill, they always tell you to use a backup method because of the fact that the antibiotic is competing with the birth control, and the birth control will not be absorbed by your body. So that is why they always tell you to use a backup method. So that is an idea of antagonists, and I actually prefer that example. But if you remember the penicillin and the macrolides, fabulous. So know the difference between synergism and antagonism in terms of antimicrobial therapies. Okay, so how do we determine when an antibiotic is clinically useful? So the idea is that you want to have a peak of antibiotic, and then eventually what will happen, so the idea of the reason why a lot of times you have to take antibiotics, like up to 4 times a day, depending on which one. So the idea is that you get a peak of antibiotic, but then slowly as your body metabolizes it around like hour 5 or so, you have minimal to no antibiotic therapy anymore. So that is why you take another dose so you get another peak. So that is the idea of a half-life of antibiotic. So half-life has to do with how long does it take your body to remove half of it from your body, either through you metabolizing it through your kidneys, or your liver is destroying it, or you actually secreting it out through feces and urine. So that is the reason why you always want to take your antibiotics on time, so you can always have this peak of antibiotic throughout the course of your treatment. Because if you don't get this peak, if you don't get multiple peaks, then what's going to happen is that the microbe is going to eventually be able to grow and continue its infection process. Okay. Oh, one other thing. Sorry, guys. So one other thing is actually this first bullet point. So I really like this first bullet point. So in order for antibiotics to actually work within the patient, it has to have a high concentration of presence in your body. So that is this peak. So that peak is the concentration of medication inside your body. And then in order for microbial growth to be either inhibited or completely stopped, or killed, you need to have that constant concentration of antimicrobial medication in your body. Okay. So now with the chemotherapy index, so this is a little bit, it's important, but I'm not really too concerned about it, but I'm just going to talk about it very, very briefly. So this is the idea of where clinicians need to keep certain things in mind. So this is where mainly on like the nurse's end and the doctor's end, this is not so much on the patient end. So this is where physicians need to keep in mind like what kind of organism is susceptible to a antibiotic, and then also whether or not if it's good to treat with an antibiotic and what kind of antibiotic, and then also being able to understand the toxicity level, so making sure that they are prescribing the right dose for the patient, because on some antibiotics or even antivirals, you don't need to take 4 times a day, you only need to take them twice. So like with antifungal medications are extremely toxic to the body, but they're super efficient. So with antifungals, the clinician should be like, oh, he has a fungal infection, he should only take an antifungal once daily. Whereas if you had a bacterial infection, oh, okay, like antibiotics are not as toxic, he can take it up to 4 times daily for 7 days. So they also have to keep in mind that toxicity level, as well as keep in mind the other drugs that the patient is taking. So that's why it's also very important to always tell your doctor if you're taking anything, because some, like even vitamins or probiotics, can produce a synergistic or an anergistic relationship between the drug that you may start taking for an infection. So that's a lot of things that clinicians need to keep in mind. So that's the idea of the therapeutic index. Okay, so with that, we're going to talk about how do actual antibiotics work. So antibiotics themselves are selectively toxic, meaning that they only will attack the bacteria. So antibiotics are specifically for bacteria, whereas antifungals are specifically for fungi, and then antivirals are for viruses. So antibiotics are only effective against bacterial infections. And there are multiple modes of mechanism of which how do they actually work, and we're going to talk about some of them, but the 6 are the most common. So there's where they disrupt cellular wall synthesis, and then there's where they will destroy or interrupt DNA synthesis as well as protein synthesis, or they will attack the integrity of the cell membrane, or they will attack the RNA synthesis and metabolism. So from there, we're going to look into some other processes. So this is kind of like the big picture of all of them. So if it attacks the metabolic pathway, then it normally will attack like an enzyme or a substrate of some sort. Now, if it is a cell wall inhibitor, meaning that it will interfere with cell wall growth or maintenance, it's like a penicillin or vancomycin. And then a cell membrane, this is where it will attack the cell membrane, not the cell wall. Not as common. The more common ones are the cell inhibitors, as well as the metabolic inhibitors. And then DNA replication, so that means it'll somehow inhibit DNA replication. Or RNA polymerase, so it will actually stop RNA polymerase from working, or either it'll somehow, I don't know the actual mechanism, but it will, I don't know if it directly competes with RNA polymerase, but it somehow will not allow RNA polymerase to bind. And then there's protein synthesis. So this is where it will actually alter the ribosome. So either if it alters the 50S subunit or the 30S subunit will determine what medication it is. So if it's a 30S medication where it attacks the 30S subunit of the ribosome, then an example of that is like tetracycline. And then if it attacks the 50S subunit, then an example is a macrolide or erythromycin. So that's just like the big picture. Okay, so now let's go to antibiotics that actually target the cell wall. So the bacterial cell wall is a very great candidate for medication because of the fact that we don't have cell walls. So meaning that medication will not hurt your patient. So it's a very advantageous drug target because we don't have a cell wall. So we know for a fact that that medication will not hurt our patient. So a lot of times what will happen is that within the cell wall, we have the NAG and NAMs. So those NAG and NAM is specifically made first in the cytoplasm, and then it becomes part of the cell wall once it crosses the cellular membrane. And then the disaccharide units are then assembled. And then what happens is that eventually at the cell wall, you get like this large, long strand of peptidoglycan with those NAG and NAMs. So what happens is that antibiotics, it will target that process. So it'll target the process of actually making the cell wall. So the process of making the cell wall is the addition of NAG and NAMs. So antibiotics that specifically target the cell wall will interfere with the addition of NAG and NAMs. And penicillin is one of them. So as we saw in the previous slide, so penicillin is one of the ones that actually will inhibit this process. And specifically what it does is that enzymes will attach to those disaccharide units, so the sugar units of the pre-existing peptidoglycan. And then what happens is that it will produce the peptide bond. It will produce a peptide linkage, not a bond, I'm sorry, a linkage. And then what will happen is that the fact that this enzyme is binding and producing these peptide linkages, there is not an intact cell wall anymore. So then growth is eventually, the cell will eventually collapse in on itself because the cell wall is no longer, doesn't have integrity anymore. Another one is can't even say it, the cellulophoreins. I'm gonna go with that, the cellulophoreins. So what happens with the cellulophoreins, it's a semi-synthetic drug, so it's not 100% natural, whereas penicillin, you can find it in nature. So this one was made more in a lab. But what happened in order to make this one is that chemists, what they did is that they modified it and they improved it because of the fact that there was microbes that were becoming penicillin resistant. So the fact that it is now modified, microbes have a harder time fighting against it, and a lot of times are not resistant to it anymore. So that's really nice. So that helps that if you have a penicillin resistant drug, we have another drug that you can give to your patient. So it's not as if they're going to pass away from this infection. Okay, so vancomycin. So vancomycin is, I believe he's probably one of the newest ones that we're going to talk about in this lecture. But he is considered a last resort drug because of, you don't want to build resistance because he is pretty new to the market. It takes about 15 years, guys, to get a drug on the market. It's crazy how long it takes a drug to get here. But you don't want to give vancomycin right away until you know that you have a penicillin or cellulophorin resistant microbe. But vancomycin's mechanism is very, very different compared to penicillin. And I don't believe I have it. Oh, I do. Okay, so the way that vancomycin works is that it will bind to a part of the peptide. So it'll bind to a part of the NAG and NAM disaccharide. And what it does is that it will block the addition of peptidoglycan. So rather than actually disrupting it, what it does is that it actually inhibits. So that is the mechanism of vancomycin. So microbial resistance to cell wall inhibiting antibiotics. There are two main ways that they have developed resistance, primarily to penicillin, and that's one way of where the first way is that they have a gene that actually encodes for an enzyme called beta galactamase. So this actually will cleave the penicillin. So the beta lactose ring is right here where this N and then there's a box and then an O. So right here, so the enzyme that a penicillin resistant microbe produces will actually cleave right here. And then with the fact that penicillin is now cleaved, it's no longer viable. Like it's no longer penicillin. So it's not doing its job. And then the second one is where when the penicillin binding protein is altered by a mutation. So with that, the enzyme will still be able to synthesize cell wall because it doesn't bind the antibiotic anymore, meaning penicillin can no longer enter the bacterial cell. So if penicillin cannot get in, then the bacteria can still produce its cell wall, and then it can still cause infection. Okay, so with that brings us to the drug target of cell membranes. So with this one, I really just want you to know that an example of it is Neosporin. So what actually happens is that it will dissolve the inner membrane by disrupting the phospholipid interactions. So with this one, it has to be topical because it can actually damage the human cell membrane if ingested. So that's why you don't ever see polymyxin as a pill form. It's only topical because of the fact that it can be damaging to human. Okay, so that will bring us to DNA synthesis and integrity. So with that, you have to be pretty careful with treating a infection using a drug that uses DNA synthesis. But the drugs nowadays, they're a lot more specialized because the genome of a bacteria, the way they synthesize, is definitely very different compared to ours. An example of that is the metroazole. So this is commonly known as Flagyl. So it's considered a prodrug, meaning that it doesn't become activated until it is metabolized by the microbe. So what happens is that if you take this drug but then you actually don't have an infection, then what will happen is that you'll just pass it through your system and you're good. But if there is a microbe, then what will happen is that it'll be metabolized by the proteins found with the microorganism, and then the drug will become active and actually start doing its job. Okay. So next that will bring us to metabolism. So these are sulfa drugs. They're pretty common, but what they do is that they will actually interfere with the synthesis of metabolic intermediates such as folic acid. So folic acid, it is a precursor to making nucleic acids. So if you don't have any folic acid, then you can't make nucleic acids. So that is actually how sulfa drugs work. So they will prevent the synthesis of folic acid, in turn preventing nucleic acids. So with that, it is interfering with the metabolism of a bacteria. So that's pretty cool. Okay, so another example is through RNA synthesis. So the most common one is rifampin. So this one will actually bind directly to the bacterial RNase polymerase, and it will prevent transcription. So if it prevents transcription, then you don't get any protein synthesis, and with that, the bacteria will no longer be able to make the needed proteins for itself, and then eventually will die off. Okay, so now we're going to be talking about like true spectrum true broad spectrum antibiotics. So these will target protein synthesis, and the main one that I want you to focus on is tetracycline, or also known as doxycycline. So this will bind to that 30S subunit of a ribosome, preventing tRNA from actually adding on amino acids to the A site. So that's one way. And then there's another one with the erythromycin, so the second bullet, the macrolides. So this one, he will actually bind to the 50S subunit of a ribosome, inhibiting the translocation of the tRNA that was added from the A site to the P site. So the image of that. So the first one that we talked about was the tetracycline. So what it does is that tRNA cannot come in and add to the A site, whereas erythromycin, what it does is that it prevents it being translocated from A site to P site. So it's a little different, but they both target protein synthesis. Okay, so how do we get resistance? There are 4 main ways. So the first way is that it will modify its target, or it'll destroy the antibiotic, it'll modify the antibiotic, or it'll just pump it out. So the first way of modifying targets, meaning that it will modify its cell membrane or cell wall composition, and with that, the antibiotic can no longer bind to it, or it'll destroy the antibiotic, so kind of like how we talked about with the beta-galactamase. So with the beta-galactamase, that enzyme, it will cleave penicillin, and then with penicillin no longer being penicillin, it's no longer effective. Or instead of cleaving it, instead of cutting it up, it can actually just add certain groups to it, so it'll just put a lot of junk onto the antibiotic, and with that, it's no longer effective because it's no longer antibiotic. Or what will happen is that it will pump it out through an efflux pump. So this is where an antibiotic, it enters, but then the bacteria is just like, holy crap, I don't want it in here, and then it will just pump it out through this efflux pump. So with the antibiotic no longer inside the cell, it can't function properly. Okay, so with that, also misuse leads to resistance, leads to mutants. So in order to avoid this, ideally you don't want to use outdated or weakened antibiotics. You also want to avoid taking antibiotics for little conditions that your body can clear naturally. Also, you don't want to take it under the inappropriate conditions, meaning you don't want to take an antibiotic if you have a viral infection. Also, you don't want to use antibiotics if they are somebody else's. Like, you don't want to use anybody else's prescription, because you might be actually taking an antibiotic that actually isn't effective against that pathogen. And then also failing to complete the regimen. You need to finish your medication, guys, because just like how we talked about those peaks, that you always want those peaks of antibiotic in your tissue. If you don't finish it, then you're no longer having peaks, but then that can also lead to bacteria getting resistant to it because it will get used to the antibiotic being present. Okay, so with that concludes antibiotics. Now we're going to go into antivirals. So with antivirals, so I'm just going to move to the next slide. So antivirals, it's very difficult to come across antivirals, but they are effective. Probably the most common one that you hear is Tamiflu. But the way that they work is that what they do is that they prevent the virus from uncoating or releasing. So Tamiflu specifically, what it does is that it's that NA inhibitor for the flu. So it will act by preventing the virus from actually leaving the cell. So what will happen is that the virus will enter the host cell and it'll replicate and everything, but what will happen is that rather than it being able to release its progeny, the medication will prevent it releasing its progeny, and with preventing releasing its progeny, it can't continue its infection cycle. Now these drugs are specifically for HIV. So There is a plethora of drugs for HIV, but the most common ones is the protease inhibitor. So this one will actually inactivate the protease enzyme of HIV. And with that, with the protease inactivated, new viral particles are made, but they're immature. And with them being immature, they can't continue infection cycle. Or there's another way by actually inhibiting entry completely, so meaning that the virus is preoccupied with the medication. So with the fact that the medication actually binds to the virus, it can't bind to the host. So the virus will never attach to the host cell. And then there's also direct HIV treatment regimens called HART. So this is where you add a combination of medications of antiretrovirals to your patient. So this is where they have a constant high activity of antiretroviral medications in their system in order to diminish the effect of the HIV virus. Okay. Now, one last thing for antifungals. So antifungals, they're pretty new to the market, relatively speaking, but there are 6 main categories. There's polines, azoles, alamines, caspofungin, gisrofluvin, and flucytosine. So they all have their different mechanisms, just like antibiotics. The biggest ones is azoles and flucytosine. So azoles specifically, they will target ergestrol inside their cellular membrane. So with the azoles, with them targeting the ergestrol inside their cellular membrane, it becomes unstable and then eventually will collapse in on itself. Or there's flucytosine, which will inhibit DNA synthesis of the fungi. So those are the big ones. And that will conclude our... Actually, one last thing. Just kidding, guys. The downside of antifungals is that they're extremely toxic to your patient. So they're very, very hard on the liver and the kidneys. So definitely be mindful of when to administer them and when not to. But now with that concludes our lecture, guys. So I know it was kind of a long one, but I'm glad that y'all sticked it out. If y'all have any questions, let me know. If not, y'all have a good night.